Okay, I think we'll start. Thanks everyone for coming. I know there's BC Cancer Research Day today, so thank you for making it. I'm Sabine. I'm the new coordinator for GCI Research Rounds, and you probably got emails from me about the podcast as well and a lot about research rounds. So that's me. And today our moderator will be Dr. Andrea Nielsen, and our speaker will be Dr. Michelle Hill. So Andrea, go ahead. Do we have a camera? The camera.
Okay, perfect. Hi there. Good afternoon, everyone. I'm Andrea Nielsen. I'm one of the gynae oncologists here at BCH and BC Cancer and a clinical assistant professor at UBC, and I'll be moderating for you today. It's my pleasure today to introduce Dr. Michelle Hill. Dr. Hill is a biomedical scientist and entrepreneur dedicated to transforming women's health through innovative biomarker panel-based blood tests, beginning with ovarian cancer. She earned her PhD in biochem at the University of Queensland in Brisbane and went on to complete postdoctoral research in Switzerland and Ireland before returning to Australia. She went on to establish a glycoproteomics biomarker research program, leading to the successful validation and out-licensing of biomarkers for esophageal cancer. More recently, she transitioned from academia to industry, completing a fellowship at the life sciences startup Microba. In 2023, she founded Proseek Bio, a company working to translate promising ovarian cancer biomarker research into clinical benefits for patients. Please join me in warmly welcoming Dr. Michelle Hill.
Thank you very much for the invite. I just want to check the sound is all okay.
Yes, that's good.
Wonderful. So when I was given the opportunity to speak to you guys, I was really hopeful that I was making a trip to Canada. So right now I'm in Montreal. I have been attending the Mass Spectrometry and Advances in Clinical Laboratories conference. And I had actually hoped to stop in Vancouver because my flight was transiting from there. Unfortunately, it didn't work out. So I'm again speaking with you remotely. However, the timing is much better because back at home in Brisbane, it's 6 a.m., whereas now it's much more civilized 4 p.m. So very nice to speak with you guys. And I hope in future there will be an opportunity to set up some collaborations and work with you as well. So here we go. So today's talk, I'd like to cover a couple of topics, a very short intro to glycoforms and the glycoproteomics pipeline, some background on the technology and the proof of concept work that we have done, which is all published, our ovarian cancer biomarker discovery work and the initial validation, and then move on to introduce the company, the startup that we set up and how we want to address the unmet needs and get some feedback from our clinical studies plan and then receive some input from you guys as well. During the talk, please feel free to ask any clarifications while I'm going. However, I can't really see, so you just have to shout and interrupt me. That's okay as well. And hopefully we'll also have time for discussion at the end. So firstly, our biomarker discovery platform focuses on glycoproteins and particularly the glycoforms. The reason for that is it is an area of biomarker discovery and diagnostic testing that's not covered in most test modalities. So there are multiple and long research implicating glycosylation changes during cancer development. So protein glycosylation is shown in this graphic. It's modified of sugar on different proteins, and there can be different types of glycosylation on proteins. It is added during the protein synthesis, so post-translational modification, before the proteins are released. So most of glycoproteins, most proteins in the serum are glycosylated. And the glycosylation modulate the function in terms of stability and interactions. In terms of cancer detection, glycoforms of tumor markers have been reported to be much more accurate diagnostic biomarkers. For example, in ovarian cancer, there were studies reporting if we monitor particular glycosylated forms of CA125, the performance for diagnosis is much better than just monitoring CA125 in total. However, there are no existing tests that monitor the glycoforms of protein. In terms of to be able to measure glycoproteins, there is a part of a translation gap from clinical proteomics. So my background is in proteomics research, and in the field of proteomics, the post-translational modifications is an area that is quite intensively researched. However, the research workflows are not currently directly translatable to the clinical labs because there's a lot of sample processing. Sometimes it becomes a bit of an art rather than analytical science. The data analysis usually are designed for sample batches rather than individual samples that comes through. In addition, there's a very low rate of biomarker validation due to the need to change in the technology or type. So just as an example, particularly focusing on blood biomarkers, which is what the non-invasive testing is what's often the ideal for screening. When one looks at the serum serum proteins, there's a massive range in the dynamic range. So the most abundant proteins in serum account for the majority of the serum protein. So using general proteomics method to identify biomarkers is very, very challenging because of these high abundance proteins that account for the majority of the proteome in the serum. And therefore, there needs to be some different approaches. So often when researchers look at cancer tissue, for example, as the start for finding biomarkers and then attempt to translate to serum assay, there's a gap in terms of the markers do not translate over. And on the other hand, also, once the biomarkers are found, there will be additional translation time between finding of the biomarkers and setting up into immunoassays. So to address this range of technical gaps in terms of from the clinical research, clinical proteomics to a clinical assay, many years ago in my lab at the University of Queensland at that time, we set up a platform that's called lectin magnetic bead assay coupled to mass spectrometry. It is quite a simple process, which I'll just briefly describe to you and then show the work that we've done in developing this and applying it for biomarker discovery. So the approach is focusing on different types of glycosylated subproteome. So in one step, we wanted to simplify the proteins that we are analyzing on mass spectrometers to reduce the problem of the dynamic range, but also to focus on the functional glycosylations on these protein subtypes. So the concept is relatively simple. So we use lectins, which are naturally occurring proteins whose job is to actually recognize these different glycan types on different biomolecules. So we use that as an affinity pull down and couple it to magnetic beads. Firstly, we reduce and denature serum to break up the protein complexes in the serum so that we just pull out the particular types of glycosylation that's recognized by individual lectins. Then once we washed off the unbound other serum proteins, we have a subproteome of serum that's focusing on particular types of glycosylation. We do a very quick bead trypsin digest without much processing, collect the peptides that are released, and then use mass spectrometry to either discover which proteins that are there. So that's using a very simple shotgun proteomics at that time. So this method has been developed over the last 15 years. And then in the second phase, we do a targeted mass spectrometry analysis to validate the biomarkers. So that is the general principle. doing this design, at the start, we were already thinking about how to develop a pipeline that has the same biochemistry, but then can be adapted easily to a clinical assay. And so we built in this workflow particular features, which includes a single step in enrichment, so the lectin pull down, but we also spiked in internal protein standards to monitor the quality and the processing. And of course, from the start, we wanted the single platform using this pull down, which can achieve biomarker discovery as well as validation. And then now with the company developing it into a product that the clinical lab can use. Because we use naturally existing lectins, we are not trying to use different antibodies, which could be difficult to develop. And then by using a proteomics pipeline, we can use a multiplex biomarker panel in a single assay, which I will show you in the next few slides. This allows us to have a higher performance in the multiplex assay rather than in a single assay. But at the same time, because we, rather than a single marker, we have multi-markers, but we're running it in a single assay, and therefore it reduces some of the operating costs in terms of considering a clinical assay. And then lastly, we also wanted to make it very easy for the clinical labs to adopt. So we designed the entire platform to be operational on existing triple, quadruple mass spectrometers that are very common in advanced or major clinical laboratories. So they don't need to make a major equipment purchase. And then with the company, we're also now developing the reagent kit that's easy for the lab technicians to run. So overall, from the start, we designed it so that it's very translational. So briefly sharing with you the papers and through the publication. So these are all published. If you're interested, I can actually send the articles later on as well. So the initial publications 2010, 2011 were just developing and describing the lectin magnetic beads. And then the entire method was published as a book chapter in 2023 with some collaborators who did tech transfer the method, and those partners eventually licensed the esophageal cancer biomarkers that we discovered using this LEMBA MS method, and they are now commercializing this into a test that's called ProMarker ESO. So now I will run through firstly the proof of concept study for the using esophageal cancer as the proof of concept, and then we'll get on to the ovarian cancer. Are there any questions so far? If not, I will continue. So the question for the esophageal adenocarcinoma diagnosis is the clinical need is that currently diagnosis is by endoscopy with tissue biopsy. So it is an invasive procedure. In Australia, that's a day surgery. You go to the clinic to have that performed. And because this is the cancer that occurs between the stomach and the esophagus, it's related with gastroesophageal reflux. So patients already experience heartburn. They're already in discomfort. Usually it's diagnosed quite late. So the goal of this project was to identify serum biomarkers which could distinguish between esophageal adenocarcinoma and its precursor condition called Barrett's esophagus, which is a dysplasia of the esophagus, as well as healthy controls. So in the project, we set up a lectin magnetic bead array, which we used 20 different lectins individually coupled and arrayed on a microplate. For this discovery phase, we had around 10 patients per group of healthy pre-cancer and cancer. We use the array to identify but candidate biomarkers and shortlisted, I think it was at that time, 3 different lectins and generated a targeted method for biomarker candidates. And then using an independent cohort of 60 patient samples, again, 20 per group, healthy, benign, and cancer, to then quantify or qualify the biomarker candidates in a second population. So that was in the initial paper. And then in the second paper, we then evaluated more patients, both in an Australian cohort as well as an independent US cohort, to ensure the finally selected candidates are applicable across different populations. And in that paper, we also selected a biomarker candidate, the top one, which was complement component 9, and examined its tissue expression. In that case, we could not look at the glycoform because there are no glycosylation-specific proteins, antibodies for immunohistochemistry of complement component 9. So it was just the tissue expression. So with that, we then had a candidate list of biomarkers, which was performing well to differentiate esophageal cancer from precancer and from healthy. So we underwent tech evaluation with another Australian company called Proteomics International, which then, after 1 year evaluation, they confirmed that it was a platform that was suitable in terms of reproducibility and stability for clinical assay. So they then licensed it from the research institute I was in at that time, QIMR Medical Research Institute, and are now furthering the clinical development. And recently published a paper on a new cohort with a final evaluation of the marker panel that they're now bringing to the market. So that's initial proof of the platform that it is capable of discovering and validating biomarkers and translating to a clinical assay. And now I'd like to move on to the topic that we're here to discuss, women's health and particularly ovarian cancer. And I think with this audience, I don't need to speak too much about the epidemiology and the outcomes of ovarian cancer. It is the most lethal women's cancer due to diagnosis at late stage. And the problem that we want to address is the lack of non-invasive testing for accurate diagnosis or screening. So we were interested to use our LEMBA-MS method to find some biomarkers. So I was fortunate a few years ago to get a Ovarian Cancer Research Foundation Australia grant to address the problem of early detection. So applying our pipeline, this was a project that we worked with the group of Professor Usha Menon from University College London on. So the same pipeline that I described before for the esophageal cancer. In this case, we focus on high-grade serous ovarian cancer. We compared 3 different groups again, so healthy, benign condition, as well as the high-grade serous ovarian cancer. So that being the most common subtype with the worst outcome. In the discovery phase, we used UK samples from 2 different studies that were provided by UCL. So the 2 different studies are the UK opiate study, which is the serum samples were collected at time of diagnosis. The second cohort we were able to receive from them was the UK CTOX study so this is a longitudinal study looking at screening of ovarian cancer. So we had samples that were collected around 12 months before women were diagnosed with cancer, benign condition, or remained healthy. And these 2 cohorts were then age matched as well. So in this study, we began the screening phase with 7 lectins and these lectins were selected from the literature. So there's a body of literature on ovarian cancer glycosylation changes. For the esophageal cancer study, there wasn't any literature, I think, at that time for any type of glycosylation studies in that cancer. So we did a broad screen. There were a lot more studies in ovarian cancer compared to esophageal cancer. So we had a literature base to start from. So from these discovery cohort with 7 lectins, we then did the LEMBA with the shotgun proteomics, then selected candidate biomarkers from both of those cohorts, comparing cancer versus healthy and cancer versus benign. We selected all of the candidate biomarkers from that were differential between those conditions with quite a lax statistics, p.01. So sorry, p-value of 0.1. Then developed a targeted proteomics assay of scheduled multiple reaction monitoring, and then validated these biomarkers in an Australian cohort of 95 samples, again in the 3 groups. We then, in the second phase, reduced the number of lectins to 3, which were the most, has the most number of candidates from the first phase. And so now I will show you some of the data that we got from this study, which is published. So in the initial look, we wanted to see what are the candidate proteins that came out from, that got validated during this cohort. So the requirements are, even though the analysis were peptide level, and we selected different peptides from each protein for the validation phase, we, in this kind of strict analysis, we required, in order for a protein to be a candidate, all of the peptides that were monitored should have changed consistently between the cancer and benign or cancer versus healthy groups. So from here, you can see the 3 lectins that were used for the validation. We had a number of peptides that were significantly different, between 38 or 47 per lectin. And then those correlated to between 19 and 23 proteins. And so if we further select that every peptide that were measured for that particular protein should be consistent, we come down to a number of candidate proteins that were selected to between 5 or 6 per lectin. And you can see the number of the names, the UniProt IDs for the proteins that were considered validated in terms of the protein level for each of the lectins there on the screen. But as I mentioned at the start, the power of this technology is the ability to make multi-marker panels in a single assay. And so we then went on in the data analysis of the validation cohort to formulate some multi-marker panels using multivariate statistics. And so in this table, we find the best performing multi-marker panels for each of the 3 final lectins that were used in the validation. So I'll go into a little bit more detail here just to explain what we're seeing. So using the AAL lectin as an example. So from the very left side, AAL is a lectin that binds to fucose sugar, glycan, on proteins. So what we're looking at here are the glycoforms of these proteins, which were modified by fucose. And on the left side, each of the rows indicate the UniProt name and the specific peptide that were chosen as the biomarker. And so then that's the same. And the percentage here, number times selected in the cell, is the number of models that selected this peptide. So for example, in the AAL data set, we had 70 samples. The first 2 biomarker peptides were selected all 70 times when we built the best performing model. multi-marker panel. So they're very, very stable peptides that differentiate between high-grade serous ovarian cancer in the 70 samples compared to healthy and benign. So we combine those two as a group to build these models. So these marker panels are potential biomarker panels that can be used to distinguish women with high-grade serous ovarian cancer compared to non-cancer. So what's really interesting is the different 3 glycan types have mostly different biomarkers. So the glycosylation changes are occurring on different proteins. You can see with the exception of the top biomarker candidate that's selected across all of them, the others are not the same. This analysis here was really, really super exciting. So always researchers, our ultimate goal and dream is to have early detection. So when you think, what does it mean? It means you want to be able to do screening tests before the patient, the women have any symptoms. So how do we find those sort of biomarkers? How do we test it? So you really need to do longitudinal collection, population study, collect the serum samples, so then we can go back to know, okay, this is the time point at which markers were changed, and can it be detected before the person even realized that something is wrong, that they have cancer. So we were fortunate to have access to the UK CTOX cohort, but we used this in the initial discovery. So here we're just showing the analysis from the 9 protein level biomarkers. When we look back into the discovery data, we found that 3, and here the screen is just showing 2 because it was too difficult to fit too many in there. 3 out of the 9 markers were actually altered already in the 12 months before diagnosis sample. So it shows potential for early detection. So that was really exciting. And on this slide, what I'm showing you is the left side is the discovery phase data from the UK CTOX. So that's the data that's collected, the serum sample that's collected 12 months before. On the right side are the validation phase data that was in samples that were collected at time of diagnosis. And we're basically looking at the trend of the 3 groups. So healthy is gray, benign is blue, and yellow is a high-grade serous ovarian cancer group. So you can see at time of diagnosis and at 12 months before, we're seeing very similar patterns, suggesting these markers can be detected very early before symptoms. Of course, we have to do much more validation results. So what are these markers, and are they consistent with what we know about the disease? So we looked a little bit into the functional enrichment analysis using string to really understand what are the different markers. So here showing 3 of the protein-protein interaction networks of the markers, and you can see they are mostly interacting with each other. And the 9 protein level biomarkers, which I showed initially, then the 6 markers, which is the multivariate biomarker panel, as well as the 3 early detection biomarkers, which is a subset of the 9 markers. What we can notice, well, maybe sorry, it's a bit busy, but what we can notice is the KEGG pathway that's enriched in all 3 of these combinations, complement and coagulation cascades. And what is again interesting is that a lot of the biomarkers from the esophageal cancer panel also belong to this pathway, but they are not the same proteins. Cancer is known to involve immune system changes. So this is suggesting that some of these changes may occur early and through glycosylation changes that modify the immune system. So this is an area that could be further investigated in terms of prevention therapies in future. But in terms of the biomarker for this cancer detection, it's just we wanted to sense check, does it make sense, the biomarkers that we're finding. So now I move to our startup called Proseek Bio. We're based in Brisbane. We're still very, very early stage. We want to take the results that we have so far and translate it to a reagent kit that can be used in clinical labs using their mass spectrometers. So our mission is to save women's lives and health choices through these accurate tests. We start with the ovarian cancer because we already have some results from my academic work. We also aim to look into other unmet needs in women's health, and we've started some academic collaborations in order to do those research. So how would it work? And this is a depiction of what we envisage the product would look like in a clinical lab. So from the patient and from the doctor side, actually, I have to say, most of our work that we've done so far in terms of the market and the patient journey is mostly based on Australia. So we'll be really interested in some feedback on how it may apply in Canada and how it may be different. So in Australia, the patient and the doctor side, we envisage them to be very, very low adoption barrier. So basically, it's just the blood test that they're already used to. So patient sees a gynecologist or general practitioner. Okay, something may be suspected. They give them a referral form to get a blood test. They go, okay, get the Proseek OC triage blood test. They take the form to a blood collection center. The serum's collected, shipped to the clinical pathology lab. The lab technicians use our reagent kit to process the serum sample, then measure the biomarker peptides on their existing triple quadrupole mass spectrometer. They then use our proprietary software to generate a score, and then the patient report is sent through their existing system to the referring doctor. The referring doctor recalls the patient to tell them what the result is. The doctor would have other clinical information that they can definitely be using to inform the patient what their recommendation is for their clinical care. So that's what we envisage how the test would work. But of course, from the research paper to what this test is, we've been doing this kind of planning work for almost the last 2 years to really work out, is it a feasible product? Will clinicians use it? Will the labs use it? Will the women pay for it? And so I would like to present what we've developed so far, and we'll be interested in any feedback. So the two tests we are in the current pipeline that we planned is firstly for ovarian cancer pre-surgical triage, which we call OC triage test. This test is aimed to distinguish between a benign pelvic mass and ovarian cancer. And the goal here is to insert into the pathway where a woman has a pelvic mass detected, generally by ultrasound, according to the Australian guidelines. And it's not clear, is this a cancer mass, which should get surgery straight away, or is this a benign mass that can be monitored over time or treated or in a more conservative manner? So we discussed with some gynecologists in Australia, and would you use such a test pre-surgery? Some of the inputs that we got were that they were most concerned in false negatives from such a test. And so in the next slide, I'll show you some of the proof of concept work we've done to address that. And so the OC triage test, what we wanted to do then is to collect additional data now to compare the performance of our biomarker panel test compared to surgery. So that's the gold standard of ovarian cancer diagnosis. In the second test, on the right side that we would like to develop, and actually when I applied for the Ovarian Cancer Research Foundation grant, what they really wanted to develop, and probably I can speak like most scientists want to develop this, is a screening test. Women, when we did the surveys with general practitioners in Australia, they do get a lot of requests from women for screening tests, but there are no suitable screening tests right now. This is a second product or goal for Proseek Bio because the clinical study that we'll need to do takes much longer, being a longitudinal monitoring of asymptomatic women. Nevertheless, we would like to proceed with this and have defined 2 high-risk groups that we might want to do longitudinal studies on. That includes the genetic risk, as well as women with endometriosis. And so the goal here is to demonstrate the biomarker panel can distinguish between healthy and benign conditions on one hand and ovarian cancer on the other hand at an early pre-symptomatic stage. So that, of course, is a much bigger clinical study and to complete. So now I will show you the pilot data we have to answer, is it feasible to achieve no false negative for a test like OC triage, which the clinicians we've spoken with so far indicate is very important as a test. And of course, they are, and I don't know if it's available in Canada, there are some multi-marker tests already for triaging of ovarian cancer, but the survey and the research we've done in the market is that they haven't received very wide adoption because the performance is not what the clinicians expected. And so this is a very important question for us to understand. Can we achieve that very high diagnostic value? So what we did was to perform a subset analysis of our published study, looking at the subset of benign versus cancer. Because remember, in our initial study, we had 3 groups, healthy, benign, and cancer. But in the case of an OC triage test, the woman is presenting with a mass. So the question is it benign versus cancer? And from the biomarker research I've done so far, it's very easy to molecularly distinguish between healthy and cancer. But benign and cancer is much, much more difficult. They're relatively quite similar compared to the healthy group. So this was an important question for us to determine. So in this subset analysis, we were able to develop a 3-marker panel from the AAL, which is a fucose binding lectin. And you can see on the top right here, we achieved an area under the curve of 0.985, which is quite nice, using this 3-marker panel. And I've put on the left here what the 3 markers are. So they also, again, belong to the acute-phase pathway, as well as, I didn't point out before, there were insulin-like growth factor pathway as well. So those 3 proteins. And on the bottom right side, just showing you the graph of how these 3 peptides change between the benign and cancer group. So the benign, interestingly, the acute phase proteins, 2 of them both increase in cancer, whereas the IBP3 peptide reduces in cancer in the middle there. So that was promising, but it doesn't specifically address, these plots don't specifically address the false negative. So for that analysis, we need to look at the distribution of the scores to see how it can differentiate 2 groups. So the left plot here shows the 3-marker score, and you can see that there's the blue and the red, so benign and cancer, are separating nicely according to score. There is a benign that is misclassified in the cancer group here. In the right side plot, we're looking at the negative and positive predictive values. We basically want to have very high negative predictive value as well as positive predictive value. So in this analysis, we're assuming a 20% clinical prevalence. So 20% of the group are cancer and 80% are not cancer, which reflects roughly the current situation where up to 80% of ovarian cancer surgeries are performed on women who didn't have ovarian cancer based on the tissue pathology. So the 3 dotted lines here show the negative predictive value at 1, 0.99 and 0.98, and the positive predictive values are all reasonable in terms of between 0.54 and 0.82. So this suggests that we can achieve high negative predictive value that's required for clinical utility using our technology. So far, this is what we have done and what we plan to do. So the research phase took quite a long time. We've now, in the startup phase, set up the patents and the commercial strategies. Right now, our company is implementing an ISO 13485 quality management system so we can develop the reagents for clinical use. So with the 2 products, OC Triage and OC Screen, we plan to next phase conduct more biomarker-based clinical studies to finalize the marker panels. And so the biobanks will include other subtypes of ovarian cancer. To ensure cancer specificity, we will also evaluate other common women's cancers, as well as a general population group to set the different cutoff levels. From here, select the final biomarker panel and develop the algorithm. In Australia, we plan to roll out the product with our pathology partners as a lab-developed test first, while obtaining regulatory approval. And we've already spoken with the Australian regulatory body, which is the TGA, to confirm what we need to do in terms of the clinical studies in order to receive approval. And we'll also be doing that with the FDA as well. I have to say, so far, I have not looked into Canada, so that could be more homework to do. So the same approach for OC Screen, which probably will take a lot longer in terms of the clinical study, as well as new diagnostic tests. So currently, we've set up 2 different studies, and we'll be interested to either get input on the study design, as well as potential clinical study sites in Vancouver. So I will describe these 2 briefly, but we'll not read out all that's on the screen. But if there's interest, I'd love to get in touch and discuss a bit more detail. We're happy to share the draft protocols that we've set up. So the first study is for OC Triage. So it's an observational trial to look at the biomarker panel that we will be finalizing in the next 6 months or so. So it's aimed for women coming through and who will be having surgery to determine whether their pelvic mass is ovarian cancer. So then we need a serum sample collected prior to any treatment. And then, and this serum sample can be the one that they are taken for example, the CA125 biomarker already. And so then the goal is to compare the surgical outcomes, whether or not it's ovarian cancer, to our biomarker and seeing how the performance is. And then for the OC Screen, this is a longitudinal study focusing on women with the higher risk for ovarian cancer, so genetic risk, as well as endometriosis patients, as I previously described. This will be longitudinal collection of serum samples and then referring to any clinical results, whether or not they were or they become diagnosed with ovarian cancer. In Australia and also other countries, women with BRCA mutations have been encouraged to have preventative surgery. So this kind of makes it slightly difficult in terms of that group of recruitment. So it will be also interesting to understand in Canada whether that is also part of the clinical practice already. So just as a summary and overall, we have a platform which is ready for clinical implementation. It looks at glycoform changes in serum proteins, which seem to be relating to the immune system modifications. So we're applying it initially to early cancer detection, focusing on women's health, but there are also other areas that we are really keen to get into in terms of immune and inflammatory conditions, which disproportionately affect women, and possibly companion diagnostics as well. So I'll leave it on this page, and I would love some input from clinicians when we discussed with the Australian regulators about the clinical study that we will be conducting. They asked us to collect some clinician preferences in terms of the cutoff points and the thresholds in terms of how they would use it. And so for clinicians involved in ovarian cancer diagnosis, if you wouldn't mind, we'd love some survey results to help us with this question. So that's the end of my presentation, and I'd be very happy to open up for discussion.
Thank you very much. That was a very, very interesting talk. I do have some questions, but I want to see if anybody in the room or anybody online has questions. Like Dr. Huntsman.
Yeah, thank you so much for the presentation. A lot has been learned in the last 20 years on high-grade serous cancer and the origins of high-grade serous cancer. And when stick lesions are removed, there's still up to a 30% chance of recurrence. So the identification of stick lesions or even early lesions would have to be the goal of any screening test. And you've studied invasive carcinomas where there, of course, will be disruption of the microenvironment, probably leading to some of the complement gene or protein enrichment and things like this. Do you have any evidence that your assay can detect in situ carcinomas?
So not directly. In the cohorts that we analyzed, there were some earlier stage, but they are not the main cancers. So in our next cohorts, we'll be looking at more of a different spread of cancers, as well as the other subtypes.
Okay, thanks.
Recurrence is a very interesting question. May I ask you whether there's a need to find biomarker-based tests for recurrence monitoring?
After stick, we don't understand what's going on, but it seems that cells can be dormant for 10, 15 years afterwards. And there's a lot of work going on to figure out which sticks may be at risk for having a clinical course like that and which ones aren't. And that isn't really understood. So there's a sense that all sticks aren't the same, and there's data from Hopkins and data from London on that, but not something which could be used as a clinical test to kind of sort them out or to triage them clinically.
So this is where we're looking for studies or clinicians with interest who could collect such samples, because as you mentioned, it can be up to 10 years. So that's would be such a longitudinal collection of serum samples and clinical data. If there exists such samples, then we can do some discovery work on that as well.
Okay, thanks.
think we have a question in the chat as well. So I'll read it out if that's okay.
Yes, please.
Says, thank you very much for your excellent and clear presentation. Could you let us know if the scoring system is unique? It is very important since the outcomes for IHC scientists face problems due to different scoring systems, resulting in various interpretations.
Yes, so the scoring system will be unique and only applicable to our biomarker panel. So the algorithm will go with the test.
I also have a question. I'm a practicing gynae oncologist here in Vancouver, and thinking of the noble, I believe the benign versus malignant. I think one of the clinical questions that would be really important, because when you incorporate your imaging, that is really telling in terms of your level of consideration or risk for malignancy. So in a lot of cases, depending on a woman's age, we wouldn't be comfortable. There probably wouldn't be any blood tests that would make me comfortable leaving some masses depending on their appearance. So one of the clinical situations that occurs to me is a younger woman with an endometrioma. And I just wanted to kind of open up questions about the differentiation between an endometriosis-associated cancer versus an endometrioma, which would be one of those things that I think would be most important, and women would want to know about, like, do they have to lose an ovary, to get a diagnosis, or could this be managed conservatively? And considering it's rare, it's a really good question. So I'm wondering if there's you're focusing on high-grade serous and some of the detection, but these rarer cancers will be harder to incorporate, I think.
Yeah, exactly. So I guess we started with high-grade serous because it's the most common, and we are aiming in our next clinical cohort to include all cancer subtypes, and we have those available from the biobanks we are accessing. But in terms of endometriosis, one of the studies we are setting up to do is longitudinal monitoring of women with endometriosis to to determine whether our biomarkers could be used as a screening test for those women for development of ovarian cancer. So that is one of the potential applications, which may be, again, challenging. So I guess as a startup company, we want to start with the low-hanging fruits. That's why we thought the OC triage test might be the easiest to go to begin with, but we do want to capture other cancer types as well. So one advantage, I guess, with our biomarker panel approach is different cancer subtypes, different ovarian cancer subtypes may have different biomarkers from the panel, but because our mass spectrometry-based assay can incorporate quite a number of peptides in the panel without too much cost penalty. So we could potentially formulate a test that could determine subtype as well as detection in the same test, but we still have to confirm that.
Yeah, it's super interesting. And even a detection test for endometriosis in and of itself would be very valuable as well.
Yeah, I believe there are quite a lot of work on that area right now as well. Yeah, and we can apply our biomarker discovery and translation method to many different applications if there are biobanked samples available to address these clinical questions.
Any other questions in the room or online?
I have a question, happy to ask, about, I believe it was the prompt study that you have, that one for screening.
Yeah.
So looking at your inclusion criteria, I think that it included adult women for that one. Just curious, a little bit more details on that. And how long you'll be, I guess, recruiting for it? Are you testing like 19-year-olds? I know that typically you wouldn't expect to receive a diagnosis until around like mid-40s, but how soon can you expect to detect these biomarkers? And would you do it annually with someone as young as 19?
So this study is for women with at a higher risk of ovarian cancer. So I suppose if, for example, a 19-year-old has already got tested to be a BRCA mutant, it could be relevant. With the so currently we're looking at two groups. I must say these are draft kind of protocols. We haven't actually started any studies just yet. So input will be really appreciated as well. So with endometriosis, currently in Australia, the average time for diagnosis is around 7 years. So they may not be really young women with those conditions as well. But that's a very engaged group of women who have already suffered a lot. Probably a lot of them don't know that they are at a higher risk for ovarian cancer. So in terms of starting a biobank collection, I think it could be okay, but yeah.
Thank you. Really exciting. It's great work. Thank you.
Somebody's raised their hand online. Go ahead.
Hi. Hey there. I'll lower my hand here. Hi, I'm Corinne Gardner, and Blake Jokes is a dear friend and mentor. And Dr. Hill, thank you so much for this talk. And throughout listening, and you're mentioning blood banks and some partnerships that you have in the UK. And I've come across and know of collaborations in the UK that are also looking at vaginal blood samples for biomarkers. And then Carly Buechling, whose name I can write in the chat, is also building an ecosystem to set up the depositories and a biobank in the UK to screen and offer a non-invasive tool for screening. So I was just wondering if you had already overlapped with her or other folks who are setting up biobanks in the UK.
Very interesting. Can you explain what vaginal blood collection? You mean menstrual blood?
Menstrual and non-menstrual for women in this age group who have abnormal bleeding when it's not menstrual, but
Okay.
recognize that but when it's hard to distinguish or discern if it's menstrual, because to your point, women who are experiencing PCOS and other irregular bleeding patterns that are ill-defined may offer samples.
Yeah. So no, we have not looked into other blood sources. Reason being existing biobanks, which we were accessing, generally have venous blood that's been biobanked. That's also a readily or already accepted sample type as well. But yeah, that's a possibility to have a look at the different blood types as well. Some of the potential clinical partners we were talking to are also interested in at-home sampling also. So again, we currently focused on bringing the first product to completion and make it available to women rather than trying to look at all these other things which could come in future.
Sure. Oh, yeah. This is great talk. I'll send you an email. So thank you so much.
Thank you. Thanks very much.
Any other questions? Oh, thank you very much for such an engaging and interesting talk. I also would love to chat with you some other time, so I'll be in contact. But thank you again.
Thanks very much for having me, and I'd love to start some collaborations if possible.
Thank you, Dr. Hill.
Thank you. Bye.